The amount of protein that a cell might make varies wildly. Your red blood cells, 80% of your red blood cells, protein, is alpha or beta globin. It's a huge amount. That's not true in any other cell in your body. So we're talking about pretty significant ranges of difference as to how much protein is made. How do things like that happen? Well, I'm going to describe the simplest and classic case of gene regulation in bacteria. And in particular, the famous lac operon of E. coli. So this was the first case in which regulation was ever really worked out, and it stands today as a very good paradigm of how regulation works. E. coli, in order to grow, needs a carbon source. In particular, E. coli is fond of sugar. It would like to have a sugar to grow on. Given a choice, what's E. coli's favorite sugar? It's glucose, right? Because we have, the whole cycle of glucose, the whole pathway of glucose goes to pyruvate, which we've talked about, and glucose is the preferred sugar to go into that pathway, okay, of glycolysis. Glycolysis, the breakdown of glucose. But suppose there's no glucose available. Is E. coli willing to have a different sugar? Sure, because E. coli is not stupid. If it was to refuse another sugar, it wouldn't be able to grow. So it has a variety of pathways that will shunt other sugars to glucose, which will then allow you to go through glycolysis, et cetera. Now, given a choice, it would prefer to use the glucose. But if not, suppose you gave it lactose. Lactose is a disaccharide. It's the milk sugar. And I'll just briefly sketch. So lactose is a disaccharide where you've got a glucose and a galactose. Glucose plus galactose equals lactose. So if E. coli is given galactose, it is able to break it down into glucose plus galactose. And it does that by a particular enzyme called beta-galactosidase, which breaks down beta-galactosides. And it'll give you galactose plus glucose. How much beta-galactosidase does an E. coli cell have around? Sorry? None? But how does it do this? When it needs it, it'll synthesize it. When it needs it, like there's no glucose and there's a lot of galactose around, how much of it will there be? A lot. Turns out that in circumstances where E. coli is dependent on galactose as its fuel, something like 10% of total protein can be beta-gal under the circumstances when you have galactose but no glucose. Sorry, lactose. When you have lactose but no glucose. Thank you. So when you have lactose but no glucose, E. coli has 10% of its protein weight is beta-galactosidase. Wow. But when you have glucose around, or you don't have lactose around, you have very little. Could be almost none, trace amounts. So why do this? Why not, for example, just have a far more reasonable, some compromise? Like let's just always have 1% of beta-galactosidase enzyme around. Why do we need the 0 to 10%? 10% is actually, extremely high. Well, so what? It's good insurance policy. So if I only have galactose, I need more. Well, but I mean, 1% will still digest it. I'll still do it. What's the problem? Sorry? Slow, yeah, but so what? I do it at a slower rate. Life's long. Why not? It has to compete. So if the cell to the left had a mutation that got it to produce 4 times as much, then it would soak up the lactose in the environment, grow faster, et cetera, and we get out-competed. So these little tuning mutations have a huge effect amongst this competing population of bacteria. And so if E. coli currently thinks that it's really good to have almost none at some times and 10% at other times, you can bet that it's worked that out through the product of pretty rigorous competition. That it doesn't want to waste the energy making this when you don't need it, and that when you do need it, you really have to compete hard by growing as fast as you can when you have that lactose around. Okay, so how does it actually get the lactose, sorry, keep me honest on lactose versus galactose, into the cell? Turns out that it also has another gene product, another protein, which is a lactose permease. And any guesses as to what a lactose permease does? Makes the cell permeable to lactose, right? Good. So the lactose can get into the cell, and then beta-gal can break it down into galactose plus glucose. These two things, in fact, both get regulated, beta-gal and this lactose permease. So how does it work? Well, let's take a look now at the structure of the lac operon. So I mentioned briefly last time, what's an operon? Remember we said that in bacteria, you often made a transcript that had multiple proteins that were encoded on it. A single mRNA could get made, and multiple starts for translation could occur, and you could make multiple proteins. And this would be a good thing if you wanted if you wanted to make a bunch of proteins that were part of the same biochemical pathway. Such an object, a regulated piece of DNA that makes a transcript encoding multiple polypeptides, is called an operon, because they're operated together. So let's take a look here at the lac operon. I said there was a promoter. Here is a promoter for the operon, and we'll call it P-lac, promoter for the lac operon. Here is the first gene that is encoded, so the message will start here, actually about here, and start going off. And the first gene is given the name LAC-Z. It happens to encode beta-galactosidase enzyme. Remember, they did a mutant hunt. And when they did the mutant hunt, they didn't know what each gene was as they isolated mutants, so they just gave them names of letters. And so it's called LAC-Z. And everybody in molecular biology knows this is the LAC-Z gene, although Z has nothing to do with beta-galactosidase. It was just a letter given to it, but it's stuck. Next is LAC-Y, and that encodes the permease. And there is also LAC-A, which encodes a transacetylase, and as far as I'm concerned, you can forget about it, okay? But I just mentioned that it is there, and it actually does make 3 polypeptides. We won't worry about it, okay? But it does make a transacetylase, okay? But it won't figure in what we're going to talk about. And actually, remarkably little is known about the transacetylase. There's also one other gene I need to talk about, and that's over here, and that's called LAC-I. And it, too, has a promoter, which we can call P-I, for the promoter for LAC-I. And this encodes a very interesting protein. So we get here one message encoding one polypeptide here. This mRNA encodes one polypeptide. It is monocistronic. This guy here is a polycistronic message. It has multiple cistrons, which was the dusty old name for these regions that were translated into distinct proteins. And so that's that mRNA. So LAC-I. This encodes a very interesting protein, which is called the LAC repressor. The LAC repressor, actually, I'll bring this down a moment, is not an enzyme. It's not a cell surface channel for putting in galactose. It is a DNA binding protein. It binds to DNA. But it's not a nonspecific DNA binding protein that binds to any old DNA. It has a sequence-specific preference. It's a protein that has a particular conformation, a particular shape, a particular set of amino acids sticking out that it can bind into the major groove of DNA in a sequence-specific fashion such that it particularly likes to recognize a certain sequence of nucleotides and binds there. Where is the specific sequence of nucleotides where this guy likes to bind? It so happens that it's there. And this is called the operator sequence, or the operator site. So this protein likes to go and bind there. Now, I've drawn this, by the way, so that this operator site is actually right overlapping the promoter site. Who likes to bind at the promoter site? RNA polymerase. What's going to happen if the LAC repressor protein is sitting there? RNA polymerase can't bind. It's just physically, sterically blocked from binding. So let's examine some cases here.